So, as many of you know, in philosophy of mind especially, there is a significant segment of philosophers who are in what you might call the naysaying business. And the idea has been really since the 1970s perhaps to say that psychology and neuroscience are completely independent. For example, that psychology is autonomous with respect to neuroscience. That kind of came unstuck in the 90s, partly as a result of the fact that neuroscience made a lot of progress that was really relevant to understanding certain psychological processes. Nevertheless, within the domain of philosophy of mind, there are still philosophers who are heavily into the naysaying business. And for them, the strategy is to find ways of saying that neuroscience can't explain certain things, that it cannot, for example, ever hope to explain the nature of consciousness, and that it cannot ever hope to explain the nature of rationality, and so on and so forth. And there's supposed to be many things that are exclusively the domain of philosophy. And now, this by no means characterizes the whole field, and some of the philosophers who are very different in this regard have been mentioned here, such as Chris Elias-Smith. But I felt especially sensitive, I think, about this when I started working with Terry Sejnowski on our book, The Computational Brain. And he said to me, you know, because he was a graduate student at Princeton, and he worked on cosmology before he moved into neuroscience. And Terry said, you know what John Wheeler used to say about philosophers? I thought, oh boy, here it comes. And he said, philosophers are like the cans, tin cans tied to the back of a car. They make a lot of noise, they don't move the project forward, and they're always behind. And now, that was a rather harsh thing. And Wheeler, I think, was a bit of a jokester. But I also realized that he had a point. And this came home very clearly to me this summer when I was at a meeting in Bergen, where a group of young philosophers wanted to say, look, philosophy sets the a priori limits to science. And in particular, we set the a priori limits to what can and cannot be discovered in neuroscience. Amazing. With regard to the question, and how exactly do you do that? I didn't get as full an answer as I think I deserved. Now, within neuroscience, I want to say a little bit about how we think about progress and so forth. And this is a slide that Terry Sejnowski and I did in 1992 for the journal Science when we wrote a paper. And our idea really was to replace David Marr's idea of levels with something that we regarded as more realistic. At the time, and this is still true, nobody really knows how many levels of explanation or of organization there are in nervous systems. But this seemed to us a reasonable approximation to get started as a working hypothesis. And our idea really was that research needs to proceed at all levels simultaneously, and that probably what will happen, and this has indeed been borne out, that discoveries about neurons will be understood and used by those working on networks as well as those working on synapses, and so that there would be this mutual giving of insight and ideas as well as mutual constraints up and down the organizational levels. Probably networks should be divided into macro and micro, or macro and micro and really, really micro, and so forth. But at this point, this is good enough. And up here you have a fairly recent development which depicts, using a new technology, major versus minor connections between parts of cortex. And this is really an extraordinary achievement, because part of what we want to know is what are the major highways, what are the minor spots, and how do the two sets interact. And so that hypothesis has been developed using tensor diffusion imagery, which tracks the movement of water in blood vessels, in neurons in the brain. This is a characterization of the olfactory bulb. And it's really one of my favorite beautiful things because the olfactory bulb is very different from what Jerry Fodor thinks about the brain. He thinks this is like a bowl of porridge. The olfactory bulb is so regular and systematic and organized, and you can study both the anatomy and the physiology of the olfactory system in great detail. And of course, there are molecules, and there has been a tremendous amount of work done at the level of molecules and synapses to understand how exactly the brain communicates information and how that communication can be modified as a result of the pharmacological milieu. Now, what's missing, I think, from our diagram, and this was early on pointed out, was something that Plotkin and Odling-Smee put in theirs, and that was genes on the one hand and culture as well. And of course, we know that genes play an absolutely essential role in the development of the brain, and that every time you learn anything, there is a structural change in the brain, and that structural change has to involve new proteins to make the stuff. And if it involves new proteins to make the stuff, there's got to be genes that are expressed. So how roughly then, given the reservations about neuroscience from philosophers, how do we think about these things? And I think we live with quite a lot of imprecision. And I don't think it's a bad thing. And very often, especially in the early days when I was working in Perry Sejnowski's lab, I would think, well, it would be better if we could get more precision here or more precision there. And it would turn out that if you just waited long enough, there'd be more data that would come in, and the precision would, as it were, make itself. And that there's an argument against getting too precise too early in the game. And that certain concepts, and I'm going to say this about explanation, certain concepts might be such that we learn to live with a kind of imprecision. And actually, this might be a good time to say why. So, of course, I was brought up in a philosophy of science tradition about what's supposed to count as an explanation and deduction and Nagel and blah, blah, blah. And then, of course, it occurred to me one day that explanation is something that brains do. They explain things. It's a brain phenomenon. And I also observed that what happens in the lab, and what happens in many labs that I would visit, is that students learn what constitutes a good explanation by, in just the same way they learn other skills in that lab. And here Thomas Kuhn was dead on. They learn what constitutes a good explanation by suggesting something as a good explanation and then having it criticized, by seeing how other people work, and so forth. And so it occurred to me that an attempt to get a precise definition of an explanation, and in particular what is a good explanation, may actually be misconceived. And I'll say a bit more about this later. What is, I think, good enough for present purposes, and this seems good enough for the purposes of the lab, is to say something like what Carl Craver, depending on West Salmon, it says, and that is, to explain something is to show how it is situated in the causal structure of the world. Is that vague? You bet. But is it good enough? Seems to be. We can sometimes demonstrate the power of an explanation by seeing whether we can manipulate in the real world. And that isn't always an option, but often it is. And testing an explanation by making a prediction, doing a manipulation, is actually very helpful. It is also significant, but I'm not giving you necessary and sufficient conditions. It's also significant when you can tie it in with other phenomena that are related, and I can't define related. Now, in this context, this is just so old, but it somehow has escaped philosophers of mind. I depend on the idea of Eleanor Roche, which was greatly elaborated by George Lakoff. And in her early work in the 60s, what she observed was that our workaday concepts and categories, the ones we live by in general, have a radial structure, by which she meant that we learn what constitutes a prototype of that category. We learn that there are fuzzy boundaries, and we learn that between the typical prototypes and the boundaries is declining similarity. And so, for example, in the West at least, if you ask an undergraduate classroom to tell you the first vegetable that comes to their mind, 90% of the students will say carrot. Other cultures may be slightly different. Now, radishes, there maybe 60% will say that they're vegetables, and others will say, nah, they're not really vegetables. They're, you know, they're just kind of put on the plate to look nice. And others, if you ask them then about, say, wild mushrooms, they'll say, no, no, they're not vegetables, but you eat them. Or they'll say the same thing about parsley. Well, they're not really vegetables, but you eat them. And so one way of thinking about these, and many other examples, river, mountain, friend, honesty, is to say that these, for this group I'm talking about anyway, these are the prototypes. These are some way out, and these are out in the fuzzy boundaries. What I, in my dark moments, think is that sometimes philosophers are really out on the boundaries arguing about whether parsley is really a vegetable. Maybe not, but it often sounds like that when they're drumming up thought experiments and so on and so forth. But what if your grandmother was tied to the railway track and it was really Kant that was driving? All right. So, just to give you another example, and also to show you that what constitutes the prototypes varies from culture, the notion of a house also has a radial structure. And so for people in Texas, that may be the paradigmatic house. For Plains Indians, that is. For the Inuit above the Arctic Circle, that's the paradigm of a house. Paul's mom grew up in one of those, and so forth. And so that just shows you that there can be variability for how to apply a category. What's interesting, of course, and I think both linguists and anthropologists are very onto this, is how despite this, we actually communicate quite well. Very interesting that we do. Now, I think that because explaining things is closely linked to predicting things in the evolution of the brain, explaining things is a brain function, it's a brain property. And I think that it is a radial, that the word explanation is a radial category. There's stuff way out on the boundaries where we don't know whether to call it an explanation or not, but the real hard-ass people don't, and the people who are a little ways further out do, and there may not be a right answer, just as there may not be a right answer as to whether parsley is really a vegetable. There isn't a right answer. And because the notion of explanation is also a social concept, in the way that, say, honesty is a social concept, you're also going to see cultural variability, and you're also going to see pushback from various individuals. And that's fine. Saying that it's a social concept just means that I might change where I put a putative explanation in the periphery or in the prototype as a function of how you and I interact. This doesn't make it spooky, it doesn't make it emergent. What it does is just put it into a context where slaving day and night to get the necessary and sufficient conditions for something being an explanation is maybe not the best way to spend your time. Now, within neuroscience, as I said, neuroscience is often accosted by philosophers of mind who say there are many things that they alone can address and that neuroscientists should stay off their turf. But one of the things that's rather interesting, I think, is that nonetheless, we have come within neuroscience to explain a number of things, to begin to explain a number of things. And by this I mean to make explanatory links between various levels, between, say, the level of behavior that we see in drug addiction and what we know goes on at particular synapses in the reinforcement learning system. We can document those changes. I think sleep turns out to be much more interesting than you might suppose. We all sleep, we all have to sleep, it's a biological necessity. And we all lose consciousness for a significant part of the life, our sleeping life. So it's a very interesting question for neuroscientists. What happens in the brain when you lose consciousness during sleep? Can we find out? And the answer is yes, actually, you can find out quite a bit about that. It doesn't tell you everything you are going to want to know about consciousness, but it tells you some interesting things. It tells you that here is an avenue of research that helps you penetrate the problem. There are other avenues of research to penetrate the problem of the nature of consciousness, focusing, for example, on attention, focusing on anesthetics, on what goes on during coma, and so forth. And so, and there are very interesting psychological experiments where you can find out what the brain is doing while the person is either aware or unaware of a particular stimulus, say a visual stimulus. And if you want the detail on that, because it's really fun, I can tell you later. So there's been quite a lot of push into that problem. And so when those people who are engaged in that encounter philosophers who say, yes, but you know, you're never going to solve the problem, it sounds funny to them. It sounds real funny to them because they think, what should I do? Should I sit you down and explain to you, you know, the whole schmear of what sleep research looks like, what the attention research looks like? Now, what should I do? I'm going to say a bit more about self-control shortly because self-control is something where interesting headway has been made. It involves both cortical and subcortical structures, not just in humans, of course, but it's been very well studied in all mammals. And self-control is an important component of free will. So to a first approximation, you can think of the notion of free will as being an amalgam, or a mishmash if you prefer, of two things. And one is the sort of factual descriptive component having to do with self-control. And the other one is the social component of whether and when it's appropriate to punish. So get rid of that little part for the moment, and let's just ask the biological question about what goes on in the brain when some animal is in control and when it's not. And I'll show you a little bit more about that. I'm just going to remind you again that the nervous system is complex. About 10 to the 11 neurons, 10 to the 15 synapses. Terry's lab figured that, roughly speaking, there's 10 to the 15 bits per second. Now, when they did this about 5 years ago, that was roughly equal to the bandwidth of Google worldwide. Kind of puts it in perspective. So when people say, oh, but the brain is just a machine, you want to say, oh, no, it's not just a machine. It is a really big, really amazing, really intricate machine. In the next slide, I just want to, this was also, this is a modification of an old slide Terry and I did, where what we wanted to do, just to kind of have a look at the field as a whole. So we thought, well, suppose that we do log size on the vertical axis and time on the horizontal axis. We could plot where the various techniques are, and that would show you where the holes are. Also, if you're interested in history, it will kind of give you a handle on what things used to look like. So, of course, in the beginning, there were only brain lesions. And then, of course, we got light microscopy, and that allowed people to know that there were actually neurons. Now, in the 50s, EEG electroencephalogram was discovered, and that allows you to put electrodes on the surface of the scalp and to get an aggregate picture of brain activity. And that turned out to be quite interesting, even though it's an aggregate, because it gives you a very nice portrait of the difference between what the brain is doing when it's awake, when it's in deep sleep, and when it's dreaming. And that was really kind of a remarkable result. Now, single unit recording really came into its own in the 50s. That meant that you could penetrate a cell and record voltage differences across the membrane, and that really gave us insights into what neural signaling was all about. Of course, electron microscopy gave us a much bigger picture of the nature of the synapse. I'm going to say a little bit about optogenetics because this is one of the things that was added, of course, to our original diagram. So optogenetics depended on the following fantasy. You know, if you want to drive a cell in the visual system, well, it's not too hard if you're in, say, an early part of the visual system, because you present a stimulus and some cells likely to respond. Similarly for the somatosensory system. But what if you want to find out things that are at a distance from the periphery? So, for example, the motor system or the prefrontal cortex. Wouldn't it be wonderful if you could open the skull and focus light on a cell and cause it to be active, cause it to respond? Because then you could track where that activity went and how a piece of behavior was produced. So the fantasy became a reality when it was thought that, look, the thing that we have in our retinas is rhodopsin. It responds to light. Suppose that there is, and this turned out to be wonderfully, luckily true, suppose there was a single-celled organism that made channel rhodopsin, so that it had rhodopsin in its membrane, and when light was shone on it, the channel opened and ions came in. Wouldn't that be cool? Well, there is a single-celled algae that has that. Moreover, it turns out, and this was also a great bit of luck, it's a single gene that makes it. All right, so now we've got what we need. And what you do now is have a virus deliver that gene, a viral vector, deliver that gene. And the way the technique has been modified and developed, you can deliver that gene to very particular cell types. So this cell type over here now has channel rhodopsin. I can shine a light on it and it will be active. It's an amazing new technique. Some people say it really shouldn't cover the whole space, but it's going to teach us things that hitherto we didn't think we could know without some new technique or other about places that are at a remove from the sensory periphery. And the obvious point, and because you all are philosophers of science, you know this, is that everything we know depends on having a technique for accessing that information. And your science is only as good as your techniques. And so the smart money is always moving to find more and better techniques. And in particular, of course, what the new Brain Project really addresses is that we want to understand quite a lot in this area here, not just anatomically, but we want to understand it physiologically. That's the area in which the microcircuitry lives. And that's what we really want to know. fMRI is just dandy, gives you big pictures, but the spatial resolution is not good enough to show you anything about the level of circuits or the level of individual neurons. So we love it, but it doesn't tell us many of the things we want to know about how circuits do their business, and ultimately all the action is at the circuit level. So what should a theory do in neuroscience? Again, this is really vague, but I think it's it's workable, it's good enough for now. We want to understand the level at which a phenomenon appears. We want to understand the mechanism by which that phenomenon is produced. Maybe comparable in power to our understanding of how an axon potential is produced. And we'd like to be able to predict new phenomenon. Now, I want to say a little bit about the background of neuroscience and some of the assumptions that we all use as we think about particular problems that we'd like to solve. And the first thing has to do with the evolution of the nervous system. And of course, the most important thing to think here, and it's interesting how often one doesn't think this way, is that what drives the evolution of the brain is whether or not improved behavior occurs. Ultimately, it's all about behavior. So motor control is where the rubber hits the road. And you can have the fanciest perceptual system in the world, but if it doesn't inform behavior, if it doesn't change behavior, you're somebody's lunch. And so the big driver is to enable a body to succeed at the what Paul MacLean used to call the 4 F's: feeding, fleeing, fighting, and reproduction. And now you'll notice, yeah, he was a fun guy, that these guys, of course, have really quite big brains, and they also have what's new with mammals, and that is cortex. These guys have overlying structures like the basal ganglia. They have a kind of oochie-goochie thing that is sometimes called a dorsal cortex. But it doesn't have this highly systematized 6-layer organization with highly specific inputs, highly specific outputs that seems sort of indefinitely replicable. So that all you have to do to get a bigger cortex is change those genes that say stop now and let them produce more, and they just produce more and more cortex. Of course, there is a limit, as we know. You can't have a head that is so big that the baby can't be born. The other sort of background point is worth remembering is that the basic function of perception is not to be beautiful and wonderful and a source of philosophical treatises. It's to inform behavior. It's to allow the animal to make better and better predictions so its behavior will be more and more successful at the 4 F's. And here you'll see, or maybe you won't, I kind of like this picture actually, but there's an owl who can be seen. But of course, part of what all mammals do and all birds do is move their head in order to be able to see that guy. So there's this lovely tight connection between perception and behavior. I used to sometimes sit in the bus and just watch people. And you do this, of course, with your own dogs too. And they're always moving. Their head, their eyes, they're scratching, they're moving, their eyes are moving, their head's moving. It's fascinating. We're moving around all the time. That's an important way we get information. We think we just open our eyes and see the world. Not so. All right. And learning. You can think of learning in a Platonic way, but in actual fact, it's a very, very cheap way, as Mother Nature figured out early on. It's a cheap way of getting adaptability. So that one thing you could do if you were Mother Nature is to make genes that would allow for behavior for every contingency that that animal's likely to make. That's a really big chore. It's so much easier to just have the animal tune the brain up to the local environment. And of course, that's what mammals do in spades, because we've got this great huge cortex that is so immature when we're born, particularly so in the case of humans. And the brain tunes itself up to whatever environment it finds itself in. And that's a huge advantage over animals that don't have a cortex. Oh yeah, so I just really wanted to show, I mean, you might think that with wolves bringing down a caribou, it's all reflex. Watch it sometime on YouTube. It's not all a reflex. Not everything that a rat does is reflex. Rats are extremely intelligent. They have all kinds of knowledge. Their knowledge is flexible. And we have to have a story of rat knowledge and rat beliefs and rat desires that does not depend on language, because so far as we know, they don't talk. So there better be a story about how their neural networks fold onto the world that doesn't ascribe to them propositional attitudes of the kind that Jerry Fodor likes. And these guys, well, they're learning to get money out of a machine. And finally, this is one thing that I remember Rodolfo Lina saying to me early on. He said, you know, people think the brain is an input-output device, and it absolutely is not. What he meant is kind of interesting. He meant that, yes, there is input, and sometimes there is output. But even in the womb, but always, there is a huge amount of spontaneous activity. So the spontaneous activity of the brain is something that we think alters the input or generates something like a desire or a goal quite independently of input. And so the spontaneous activity of the brain, and there's a huge amount, even when you're asleep, even when you're in coma, there is spontaneous activity. Has to be taken into account. And just for comic relief, because you're probably getting tired of this, I think that the Gestalt guys actually realized something important that relates to this notion of spontaneous activity. What you are instantly aware of when you see something like that, for example, is not a whole lot of little distinct things, and then you say, oh, I think I'll infer that this constitutes a whole. You see it as a whole, right? You see it as good continuation. You see that as symmetry. But the next one, I think, oh, I do hope this works. Yeah, it should work. Now, the next one is going to show you how, by virtue of internal activity, the brain contributes. And here I think Kant was sort of right, you know, about the brain or about the mind being constructive. The brain with kind of minimal input gives you a big story. All right, so here you go. I like this. Okay, go, guy. No! All right, so what's happening here?
It says it can't find the...
It can't, okay, well then that's too bad. Anyway, those things move. I put the movie in there, so I don't know quite why it didn't. Anyway, those things move, and you see a man as doing particular actions. It's not nearly as much fun if I just tell you this. So now I want to go back and talk a little bit about self-control, because I said that that was one area. All right, I think I've completely gibbled up your device here. Oh, now I totally have. It's all coming apart. All right, so I want to say a little bit more about self-control. I want to just give you a feel for what we have kind of picked up in neuroscience that I think might give you a feel for why. Yeah, I don't want to even touch it. That's good. All right. So in the early 1980s, Walter Mischel was a psychologist at Stanford, and he did this very famous and very wonderful experiment. He was interested in self-control, and he wondered if little kids could defer gratification. So you get your little kid, he's about I think they were 3 years old or maybe 4 years old. You get your little kid in the lab, and you put a marshmallow in front of him, and you say, you can have that marshmallow now, or if you wait 5 minutes, I'll come back with 3 for you. Very simple. And he ran it with a whole lot, let's say 50 kids. And what he found was that some could and some couldn't defer gratification. And he ran it again, and it was the same ones who could and the same ones who couldn't. And so his lab tracked these people over their lifetime, and he found that the people who could defer, the little kids who could defer gratification were the kids who grew up to go to college, have stable marriages, have a job, and so on and so forth. And it was much less true of the little kids who couldn't. Now, this is likely not to be unique to humans. And everybody who knows anything about wild animals knows that especially predators have to exercise self-control. They can't just, run after their prey and hope to get it. They have to they have to bide their time, be very stealthy, and so forth. So people began to think about the notion of self-control as involving the capacity to defer gratification, to maintain a goal despite distractions, to suppress unacceptable impulses, especially, for example, within a group, and you see that very clearly in wolves and baboons and chimpanzees and, yes, humans, and to stop an action that you began when you see it's got to be stopped. It's called action cancellation. So Trevor Robbins in Cambridge thought, okay, I'm going to try this with rats. And the first thing he wanted to try was deferred gratification. So you get yourself a feeding tower, and when the rat comes into the space, he can poke his nose on a lever and he can get a pellet. Well, they learned that real quick. Now you're going to put in 2. They're going to look identical. And in the second one, when the rat pokes its nose, it'll get 8. Big deal. Rats always go there. Now, here's what you can do. You can find out if a rat can wait for the higher payoff. So you introduce a delay between the nose poke and the goodie. Some rats can wait, and they just sit there. They nose poke, and some can't. Very interesting. So some can defer, and what I think is very interesting is that any rats can defer gratification, because there is this prejudice about that we're the only really smart guys, we're the only ones with morals, we're the only ones who have free will, and so forth. Yeah, rat can defer gratification. So then the question that Trevor Robbins asked was, well, can they do action cancellation? And essentially, what the rat would do would be he'd be led into the area, come along, and at some point as he approaches the feeding tower, a tone sounds. And when the tone tells him to stop, you train him up so that he does that. So he comes in, tone sounds, he stops. And you take him out, now we do it again. Now you can let him get closer. Can he stop? Some can, some can't. Now you let him get real close so his nose is really right up there. And you sound the tone. Some can move back and some can't. Now that's very interesting because action cancellation in humans has been studied. And guess who cannot do action cancellation? Addicts. They get close. I mean, it's not cocaine that they're being given. It's just a sweet. They can't stop. So that's kind of an interesting result. And then Trevor Robbins asked this question. What's the physiology here? Is the capacity to defer gratification the same capacity as action cancellation? Is it the same circuitry? It turns out it's not. So I'm not going to go through the details of this. Suffice it to say, well, there's a rat. But what you can see is that they propose, at any rate, that this circuitry is critical, and it involves the reward system. The nucleus accumbens, the shell, the nucleus accumbens core. This circuitry looks like it's the important one for stopping impulsivity. So for those of us who've always been a little sort of wary, shall we say, of folk psychology, it looked like an interesting case where you might have thought that self-control was a single, all-encompassing capacity, but it turns out that it diverges. Now, you're going to want to know, are the rats who could not defer gratification the same as the rats who were bad at action cancellation? No, not necessarily. There was some overlap, but some rats were good at action cancellation, but bad at deferring gratification, and so forth. It's a very, very interesting result. Now, put in the context of other things that we know about addiction, it turns out, I think, that we can see ourselves as making progress, one might even say reductive progress, on the question of self-control and addiction. Now, this is a schematic of a rat brain, and what is highlighted is the reward system. The ventral tegmental area is particularly important. You also saw in the last slide that the nucleus accumbens is an extremely important part of the reward system. And yes, you have it, we all have it, or at least we hope we have it. Two things are highlighted here. One is the dopamine projections from the ventral tegmental area forward. And those are absolutely crucial for learning all kinds of things. And in particular for learning things like self-discipline, but for learning many things. And it's that system that is in some ways corrupted, but not only that system, corrupted by things like cocaine. And incidentally, the changes made by cocaine to the human system and to the rodent system are, as far as we know, permanent. So it's not, it's not just like a joke. You will also be interested to know, for those of you who sneak out there and have a smoke, that here are the nicotine receptors in the reward system. That's why you like it. We also know that when we put people in the scanner who are smokers, and they've just had a smoke and they're not craving, and we give them a certain task, that they are poorer at that task than normal subjects. It's a I was very surprised. I thought nicotine was more benign than that. You will also see opioid peptides, the endogenous opioids that our brains make. And there are, of course, cannabinoid receptors. That's why smoking grass feels good. And the changes that happen to this circuitry have now been really fairly well documented in rodents. I mean, rodents really like alcohol, and they really like cannabis, cocaine, all kinds of stuff, just the same as humans, because we've got pretty much the same system. I mean, this is a subcortical system. Your cortex is a lot bigger than that of a rat. But, your cortex is essentially the same as a rat. Here's something I think lots of people don't know, and that is that your brain has no structure, no organizational component that a rat does not have. You got a lot more neurons, and we don't know how it is that more gives you things like the capacity to see so much further into the future, or the capacity to acquire language. So that's a big unknown. And it's not just that we're big rats. Our brains are different, but not by virtue of having different structures in them. There's no neuronal type that a rat has that we don't have, and vice versa. All right, so just still on the topic of addiction, I should wrap up soon because I realize that everybody's getting tired. So when you take the shot of cocaine, what happens is your dopamine goes up. That actually creates structural changes in the striatum, which is a subcortical structure. Opioid peptides go up, so does serotonin, and so does an inhibitory transmitter called GABA. Nobody really knows why. We notice that when people are in withdrawal, some very interesting things happen. Dopamine levels plummet, opioid down, serotonin down, GABA down. And what isn't here that we also notice that really helps explain withdrawal is that stress hormones go up. So when this happens, when GABA goes down, stress hormone levels go up. That makes people feel anxious. Anxiety is really unpleasant. How do I get rid of this unpleasantness? I take another hit. And then, of course, as you know, it snowballs. Now, I'm going to say a couple of quick things about consciousness. Just because so many people in philosophy seem so convinced that consciousness is just beyond the reach. All right. So there's many, many ways into the problem, as I indicated earlier. It all involves figuring out what techniques you can use to make progress, and that's what I'm interested in. Here is going to be a contrast between wakefulness and unconsciousness. The unconscious state is produced by using the drug propofol, which is a nice drug if you ever have a colonoscopy or something like that. That's what they give you. It's very nice, and you're out and you wake up quite quickly. So what you want to do is contrast, for example, in A, what the levels of activity high is in the white, yellow, low is in the red. And you can see that there's quite a significant difference, for example, in that cut. You can see that the more frontal medial structures are far less active in the unconscious state. And you can see a similar thing. I don't want to go into great boring detail, but you can see the contrast, what's happening there. Does that link up to any other things that we know about the role of frontal structures? And the answer is yes, it does. That this is highly schematic, but it gives you a feel nonetheless that this strong pathway between the medial, frontal, and the posterior regions is downregulated in deep sleep. That's pretty interesting. And then we can investigate what that does in terms of experience and so forth. And in this last slide regarding sleep, it's kind of a mess, but I wanted to show you this slide because it shows really that many, many, many levels can be addressed with regard to one and the same phenomenon. So we're interested in this case, in the difference between being awake and in being in the various dream stages. So we're going to use, first of all, single cell recordings in the frontal part of the brain. And what we see is action potentials. And then that changes when you go in. You can see the lack, the drop-off of action potentials. You can use extracellular recording and see what kind of effects you're getting. You can use EEG. Now that was, as you'll probably remember, in the 1950s, that was the first and only way of kind of accessing what was going on in the brain without, doing an autopsy. And it's lovely to see that the generalized waveform for being awake looks like that. For REM, early REM and late REM, it looks like that. But for intermediate REM, it looks like that. And so many people were quite stunned, as I think we all should be in a way, by the fact that REM sleep and being awake had, at this aggregate level, a very similar waveform. Now, what you also measure then is with the electric oculogram and electromyogram, you're measuring activity of muscles. Much more difficult to do is to find out what's happening at the level of the neurotransmitters. And what we see basically is that in the awake state, serotonin goes way up. In the REM state, acetylcholine goes down, dopamine goes down, and in the middle REM sleep, serotonin is up and the others are down. Now, there's much more that's been done on sleep than just this, but this will give you a feel, at any rate, for how it is that some neuroscientists are quite optimistic about making some progress on the problem of consciousness. And I think their idea is a bit like what it's like when you're hiking. You don't really know what's going to be over that mountain until you get there. So, you get there and then you look around and think, oh, I see, okay, well then I got to do this. And I think that's the sort of stage that we're in at this point. We don't really know whether we're going to get anything that will satisfy anybody, even ourselves. But we do think that it's worth mucking on. And I think I'll leave it at that. And if there are any questions, I would, of course, be more than happy to address them. Thank you.